Hello everyone. Today we'll be discussing on the types of vectors that are used for gene cloning, experiment to clone foreign gene or gene of interest for constructing recombinant DNA molecules. Vector basically they are DNA molecules which are used to transport DNA sequences between the biological host and the in vitro condition test tubes. So these vectors are what are routinely used in the plant genetic engineering or genetic engineering laboratories are genetically engineered strains, and they are not natural strains which we use for construction of recombinant DNA molecules. So basically a vector is to amplify a cloned DNA into many copies by expressing them in the biological host system. Let's see some of the properties of vector that can be ideally used for gene cloning purposes. So these vectors which we choose for gene cloning should be capable of autonomous replication in at least one host organisms. And these vectors must be smaller in size that can aid in easy manipulation or DNA manipulation techniques can be deployed for smaller size vector compared to larger vectors. And they also help in reducing the complexities associated with analyzing the recombinant DNA molecules. And these vectors should be capable of amplifying the cloned DNA sequences in many copies or multiple copies. So high copy number will facilitate maximum expression of the foreign gene in the host systems. The vector also should possess a unique cleavage site or a polylinker or a multiple cloning site for an array of restriction enzymes. So that gives a choice for any user to choose an appropriate enzyme to cut open the vector that can be used for cloning purposes. But we have to ensure that occurrence of multiple cleavage site will reduce the likelihood of the function of the recombinant DNA formation. Next, these vectors should possess one or more genetic markers that would enable them for selection of the clones. After construction of recombinant DNA, we will be going for screening the recombinant DNA molecules. So these vectors should have some selectable marker systems for easy screening purposes. And also this vector should permit detection by any simple genetic test for the confirming the presence of passenger DNA or foreign DNA molecule in that. Also, these vectors should process appropriate transcriptional and translational signal, which need to be located outside, which not outside, should be located adjacent to the cloning site for better expression of the cloned DNA sequence. These vectors should have host specificity when there is an issue of biological containment arises. So host specificity will prevent this vector escape from any biological host system to another system. Now let's see the variety of vectors that are being routinely used for gene cloning purposes. The first and foremost will be the plasmids, which are widely used for gene cloning purposes. Other than plasmids, we have phages, cosmids, phosmids, some shuttle vectors for shuttling between the host systems, expression vector to produce a recombinant foreign proteins in the host systems. And also some of the chromosomes, artificial chromosomes are modified to be used as a vector to have a larger insert capacity, which includes the bacterial artificial chromosomes, yeast artificial chromosomes, and the P1 phage based artificial chromosomes. We'll also discuss on the binary vectors and co-integrate vector systems as a special strategy for plant genetic engineering. First type of vector we will discuss is plasmids. Plasmids are basically extra chromosomal DNA which are capable of self-replicating. They are double-stranded DNA which are maintained in the bacteria, and some of the archaea also possess these plasmids. These molecules are capable of autonomous replication in the sense they don't depend on the chromosomal entities for their replication, and they are also observed in the yeast systems. And we also have a kind of plasmids which are present in the RNA. That's an RNA analog similar to the DNA plasmids present in the bacteria and yeast system. But these RNA plasmids are non-infectious extrachromosomal linear RNA molecules, and they are available both as an encapsulated as well as unencapsulated forms found in some of the fungi as well as in various other plants species from algae to the land plants or angiosperms. The term plasmid was introduced by Joshua Liederberg in 1952 for referring any chromosomal, extra chromosomal hereditary determinant. So here you can see the picture representing the double-stranded circular form of a plasmid. This image shows the electron micrograph of a plasmid, which exists normally in 3 different conformations, that is supercoiled structure, open circular structure, or linear molecule. And here you can see the plasmid, which is in a very relaxed state observed under electron micrographic conditions. Some of the plasmids normally they are observed to be integrated with the chromosomal DNA, but some plasmids may remain separate. If the plasmids do not get integrated with the chromosome DNA, we call them as non-integrating plasmids. Here, typically you can see they are non-integrating plasmids where the chromosomal, once the bacterial cell undergo binary fission, there will be a separate replication of the chromosomal component of the bacteria as well as the plasmid replicate separately because they are not integrated with the chromosomal DNA. The plasmid is not integrated with the chromosomal DNA. Whereas in the other case, you can see typically the plasmids are integrated with the chromosomal DNA, and we call it them as integrated plasmids, or they lie over the chromosome. We refer them as episomes. So the term episome was proposed by Francois Jacob and Elie Woolman in 1958. Let's see the general structure of a plasmid. A very popular general structure of a plasmid is represented here. This is a plasmid which is designed by 2 scientists, Bolivar and Rodriguez, which is by nomenclature represented with a small p and BR representing Bolivar and Rodriguez, and the number 322 representing the number of the strain, number of the plasmid that was created by these scientists. So these plasmids do possess genes for some of the antibiotic resistance, like tetracycline here, and they possess resistance for ampicillin antibiotic. Normally, for autonomous replication, a plasmid should possess an origin of replication, which will make them independent entities to replicate without depending on the chromosomal components. These plasmid vectors are basically used to clone DNA, but normally the DNA that can be cloned using a plasmid vectors are very small in range. It can range from 100 kilobase pair to 10 kilobase pair in sizes. And what we prefer to for cloning purposes include some of the features which we see in a modern day plasmids used for genetic engineering is that ideally they should be smaller in size, should possess an origin of replication ORI sequence, and they should have a multiple cloning site MCS or a polylinker region which have an array of array of restriction enzyme that will give you a choice for the user to choose an unique cleavage site cut with an appropriate restriction enzymes. And we have discussed an ideal properties of vectors, which also said that it should have a selectable marker gene to screen the recombinant DNA molecule once the cloning is done. And some are expression vectors. Plasmids can be deployed for expression purposes to produce the recombinant proteins of the cloned gene, and they have sequences that will allow transcription of the foreign DNA by using the RNA polymerases, and that can be translated to produce the foreign protein. Let's see the example of a typical plasmid with 3 basic features. First, you can see here the plasmid possesses an origin of replication, which is recognized by the host cell so that it is replicated along with the DNA that it carries. Means the DNA that we cloned in the unique cleavage site after opening, the foreign DNA cloned will also get replicated along with the origin of replication replication process. Secondly, you can see that the bacteria plasmid should possess a selectable marker that will help enable the vector to be selected or identified in a selection system to differentiate a cloned DNA from an uncloned DNA or a self-ligated DNA molecule. Third, you can see a cloning vector needs a unique cleavage site or a single cleavage site among the host of or an array of restriction enzyme, each of which will have one or more restriction site. One or more restriction enzyme can be deployed. Here, typically, you can see example with use of Hindi3, EcoR1, SAL1, BAMH1, or PST1, but we have to ensure that the restriction enzyme host recognition sites, template recognition sites in the vector is not present anywhere outside the unique cleavage site or MCS region. So here you can see the bacterial plasmid with the ORI, that's an origin of chromosomal replication that will be deployed by the cellular DNA replication enzymes to copy the plasmids. And I have already told you that it is independent of the replication of the chromosomal component. Here you can see the MCS are a polylinker region. MCS is also called as a polylinker region, which have a choice of restriction site for cloning the foreign DNA. Here you can typically see the plasmid vector is being cut open with the any one of the restriction enzyme, which cleaves them to produce a nicked plasmid DNA. So your template foreign gene can also be subjected to treatment with the same restriction enzyme, which is used to cut the host vector, and they can be ligated together to produce a recombinant DNA molecule. So these plasmids can be classified based on several basis. In the first basis of classification is based on the copy numbers. So based on the copy numbers, the plasmid can be classified into a high copy number plasmid or a relaxed plasmid, or it can be classified as a low copy number plasmids or stringent plasmids. Means the number of replication that could put forth is limited due to the stringency exhibited by the bacterial system. The second classification is based on the host range, which allows the plasmid to be propagated. Some may have a narrow host range and some may be having a wide host range. Based on the occurrence, the plasmids can be classified into naturally occurring plasmid, and some are synthetic plasmids which are constructed in the laboratory or designed by the scientists. So based on the specific function or role played by the plasmids, we can classify these plasmid as col plasmids, resistant plasmids, fertility plasmids, virulent plasmids, or degradative plasmids. In case of col plasmids, they contain the bacteriocins, otherwise also known as colicins. These are all proteins that will kill other bacteria and help to defend the host bacteria from their challenging enemies. So they are called as col plasmids. Col stands for colicin or bacteriocin. The second type of plasmid with a functional role is the resistant plasmids, which are also called as R plasmids, that helps to transfer through conjugation, and they contain genes that will help a bacterial cell defense against some of the environmental factors, xenobiotics or poisons, toxins present in the environment. The third type of plasmid is fertility plasmids. These are also called as F plasmids, which fall under the category of conjugative plasmids. They contain the transfer TRA genes, which allow the genes to be transferred from one bacteria to the other. Say, for example, the F minus bacteria can receive the genes transferred from the F plus strain through conjugation process. The fourth functional role played by the plasmid is virulence, and they are called as virulence plasmids. A virulence plasmid turns a host bacterium into a pathogen, and they can be a causative agent for causing a disease. In, say, for example, in plant species, we have a bacterium by name Agrobacterium tumefaciens, as well as Agrobacterium rhizogenes, which causes the diseases like crown gall disease. Agrobacterium tumefaciens causes crown gall disease, which possess the TA plasmid, tumor-inducing plasmids. The TA plasmid is about 200 kilobase per plasmid with the virulence regions present in that. The last classification here is the degradative plasmids, which helps the host bacterium to digest the compounds that are not commonly found in the nature. Say, for example, some of the bacterium like Pseudomonas putida possess the plasmids which can be capable of degrading the foreign compounds or the xenobiotics like oil spills. So Pseudomonas putida can degrade the oil spills or hydrocarbon compounds like camphor, xylene, toluene, and salicylic acid. This is an example of a F plasmid or a fertility plasmids, which aids in transferring the fertility factor from the deficient to from a donor to a recipient strain through the a sex pillay or F pillay that establishes the contact between the two strains. This image represents the virulent plasmids from the Agrobacterium tumefaciens that causes the crown gall diseases. This disease is very common to the palm fruits, or most of the dicotyledonous plants become vulnerable for infection by the Agrobacterium tumefaciens. This TA plasmids, TA stands for tumor-inducing, which possess the virulence regions within the plasmid region that makes the plasmid and the host a pathogenic strain causing the diseases. So if the pathogen causing part, that is the oncogenes, are removed from the TA plasmids. The oncogenes include the genes for auxin, cytokinin, and opines. If they are removed from the transfer DNA or tDNA regions, they can be deployed for plant genetic transformation purposes that can be inserted with the foreign DNA or a gene of interest that would serve as a vector system for producing transgenic plants through the Agrobacterium tumefaciens, and for which we use normally a disarmed TA plasmid vector. A disarmed TA plant vector is non-pathogenic, but still possesses the virulence factor that can help in transfer of a gene from the tDNA host plasmid cell to the plant systems. So normally, why do we choose this kind of virulence plasmid like a TA plasmid for transgenic transformation is that naturally plants do not have naturally occurring plasmids that can be efficiently used to modify to transfer the genes across plants. Unfortunately, the large size of the TA plasmid, which is around 200 kilobase pair, is making the size not amenable for manipulation. That's why there are two solutions to solve this problem. One is that we have an issue with the plant system that's not possessing the plasmid. Second is that the naturally occurring TA plasmid of the agrobacterium is large enough that is difficult to manipulate. So there are two solutions to this, and they can be adjusted to shoot for plant genetic transformation purposes. The first strategy is going for binary vector systems. The second one is co-integration vector strategy for cloning the DNA and express them in the plant systems. The next functional role played by the plasmid we have seen is the degradative plasmids, which contains genes that could degrade the oil spills or the xenobiotic or hydrocarbon compounds. For example, the CAM plasmid is present in the Pseudomonas putida that will encode enzymes that will degrade the camphor compounds, and similarly oct plasmids will degrade the octane compounds in the hydrocarbons, as well as the xylene and toluene can be degraded by the xyl plasmid, and NaH plasmids will degrade the naphthalene compound, and salicylic acid can be degraded by the sal plasmid. In the binary vector category, there are two plasmids which can be used as a vector for inducing genetic modification. So for optimally making genetic transformation, we will be cloning the gene of interest in the transfer DNA regions. So any genes that are put between the border regions, left and right border regions on a plasmid, will be transferred to a plant upon infection by the Agrobacterium tumefaciens. So in this case, typically the tDNA, which is present on a smaller plasmid, is of a range 20 to 30 kilobase pair, and the virulence and host specificity region, they can be made to be present on another plasmid, which was of size 170 to 180 kilobase pair. So practically, we are separating the approximately 200 kilobase pair size of the TA plasmid to be split into two different plasmids. One is carrying the tDNA alone of 20 to 30 kilobase pair. The other one will carry 170 to 180 kilobase pair of the host specificity and virulence region. So the tDNA of 20 to 30 kilobase base pair can be easily manipulated to hold our gene of interest, and they can be easily integrated by using the cloning process, the routine cloning process. When both these plasmids are present in the same Agrobacterium tumefaciens cells, they complement each other. Means the tDNA will complement the host and virulence factor, and host and virulence factor will do otherwise, vice versa. And the genes on the bigger plasmid are also complement for expression. So they will help to encode the proteins that are used by the smaller protein to get integrated into the plant chromosomal DNA. That's how we can efficiently transfer the gene by using a binary vector strategy to possess the tDNA in one plasmid and the virulence and host specificity region in the another plasmid. So the gene of interest also get integrated into the plant's DNA, and desired transgenic characters can be achieved by using the binary vector strategy. So here you can typically see the picture representing the TA plasmid, which is having a helper DNA TA plasmid with a virulence factor, and the other plasmid is a binary vector which processes the tDNA, where you can optimally clone your gene of interest in the tDNA part. So when this disarmed TA plasmid is cloned with the foreign gene of interest, the virulence factor here can complement the TA plasmid, disarmed TA plasmid, and helps to integrate the cloned DNA into the chromosome of the transferred or transformed plants. The second strategy for solving the problem of the larger size of the plasmid as well as the plants not naturally occur producing having plasmids is through co-integrate vector strategy. Co-integrate vectors are produced by integrating suitably a modified E. coli plasmids which will contain the gene of interest into a disarmed plasmids. So in case of co-integration of two plasmids, this is achieved by a process of homologous recombination. So for a homologous recombination to occur, the E. coli plasmid as well as the disarmed TA plasmids must have the same sequence, some sequences which are common to both for the recombination or the homologous recombination to occur through the co-integration vector strategy. So a plasmid vector is typically used to clone the DNA fragments of up to 15 kilobase pair. If you want to clone a longer length of DNA, we can aptly go for other vector systems like lambda-phage DNA or Cosmids or artificial chromosomes like BAC, YAC or PAC vector can be used for cloning the larger size of DNA. So in this case, when we go for a co-integrate vector strategy, you can typically see here the E. coli possess the tDNA, which is integrated with the a tDNA region is present in the E. coli plasmid, and also you have an integrated gene of interest in this. When this is going to be mixed with the TA plasmid that possesses the virulence factor and the host specificity region, when you recombine them, and these two will complement with each other and produce a recombinant TA plasmid, where the tDNA region has now the gene of interest transferred from here to the tDNA region. You can see a tDNA that is now encompassing the foreign DNA or a gene of interest expressed in that, along with the virulence factor and the host specificity region. So you can co-integrate the genes available from two different plasmids, the E. coli plasmid and the disarmed TA plasmid, recombining them to produce a recombinant TA plasmid. Now all the three important regions, that is the virulence factor, the host specificity factor, as well as the gene of interest is present within the tDNA region by co-integration strategy. The next vector system we will discuss is the phage or viral vector systems, which are viruses that eats bacteria. So enterophage bacteria or a lambda-phage or a coliphage bacteria, virus is the virus that eats bacteria, or they kill bacteria, and we call them as bacteriophage, which infects the bacterial species like E. coli. And this was discovered by Esther Liederberg in 1950 when she was working with the mixed cultures of E. coli strains. She could observe that some of the strains which are treated with the UV light was nibbled and plagued. That shows the plague formation due to the infection caused by the viruses that eats the bacteria, causing the plagues in the cultures. So bacteriophages are a group of viruses that use bacterial cells as a host, and they reproduce by infecting these bacterial cells. Like all other viruses, the bacteriophage are capable of producing a protein coat that will protect the genome of the viruses. So most of the phages are DNA viruses, and they code for several genes which are responsible for the viral replication. Some of the examples of the bacteriophages that are deployed for cloning purposes include the lambda-phages, M13 filamentous phages, T4, T7 phages, and P1 phages. But we'll be discussing more about a specific phage, that's lambda-phage, that's deployed for plant genetic transformation. So when we see the pattern of infection that is caused by these viruses upon bacteria, this bacteriophages needs at least 3 steps to complete its lytic cycle. So basically there are 2 types of life cycle for these viruses. One is what we call it as lytic cycle, where they make lysis of the host cell upon infection and they get released after multiplication. The second one is lysogenic cycle, where they remain latent and without causing any infection, but they still multiply within the host cell. So different bacteriophages needs minimally 3 steps to complete its lytic cycle. First, what they do is they attach at the site of bacteria, on the host surface of the bacteria, the viruses get attached, and the phage is then slowly injected into the the viral DNA into the bacterial host cells, and the genes that are present in the DNA of the phage will encode proteins or enzymes that is capable of replicating the viral DNA, the phage viral DNA inside the host bacterial cells. And finally, the other genes that also encode proteins needed for the formation of capsid, the protective layer of the virus, that is capsid formation, and the new phage that is synthesized will get assembled inside the capsids. Then finally, this third step will be followed by lysis step, where the bacterial cell, after amplification of the viral DNA inside, will release the phages from the infected cells so that they can propagate to other cells for infection. In some of the phages, the phage DNA gets incorporated into the host genome after entering the host cell, and they remain there for several cell divisions without causing any cell breakage or cell lysis. And this is what we call it as quiescent stage or a prophage stage. So the bacteria which carries the phage DNA, even after challenged by the viral phage DNA, they are not lysed, but they still remain as same as like any other uninfected cells. And this is what we call a type of infection as lysogenic cycle of bacterial viral infection. So the phage DNA finally is released from host cell by entering into the lytic cycle. So although it is a lysogenic strain which remains latent without causing any disturbance to the host cell during the multiplication cycle, they finally undergo a change from the lysogenic to the lytic cycle and get released from the infected cell to cause infection to the other healthy bacterial cells. So the infection cycle of lambda phage is an excellent example of a lysogenic cycle. In case of cloning, the lysogenic cycle is most important because the phage DNA can carry a specific gene that is incorporated into the host genome. As a result, this genome can hold good for coding some enzymes that are carried by the phage. Therefore, this lambda phage, being an example of a lysogenic cycle follower, can be an ideal choice to be used as a cloning vector for cloning larger DNA fragment size of, say, for example, approximately 23 kilobase pair of DNA can be cloned in the bacterial phage viral vector systems. Once the bacterial phage fragments are ligated into the foreign DNA fragments, they can be resulting in production of a recombinant DNA, and they can be packaged under in vitro condition, or what we call it as in vitro packaging. So in vitro packaging is about mixing the recombinant phage DNA along with the crude bacterial cell extract, which will contain all the proteins that are needed for assembling of the bacteriophage to complete a viral DNA. So the subsequent transmission of the recombinant cell DNA into the E. coli cell is highly efficient when we use the in vitro packaging system for viral DNA to be used as a vector system for larger insert cloning. So here, this picture represents the phage, lambda phage DNA. You can typically see the head of the virus, which encompasses the phage DNA, and there are head proteins as well as tail proteins present. If you see the regions here, between the regions, after head and tail, there are regions called as non-essential regions. So manipulation of this non-essential region is absolutely not going to reduce the infectivity that can cause for transmission of the foreign DNA. That's why any manipulation can be attempted to remove this non-essential region. Instead, those places can be replaced with the gene of interest used for cloning. So the approximate length of this phage DNA is around 49 kilobase pair. So there are major chunk of regions as a non-essential regions that can be removed. That's about 23 kilobase pair, can be 20 to 25 kilobase pair can be removed and inserted with the foreign DNA that can aptly be used as a vector system for cloning. So here you can see the lambda phage is resting on a bacterial host system. The lambda DNA molecule has a central gene cluster, or the region which contains the non-essential regions. So you can use an appropriate restriction enzyme to remove the central regions because they are non-essential regions, and removal of them will not cause any reduction in the infectivity of the bacteriophage system. And these regions can be replaced with the foreign DNA or insert, and they can be ligated with the ligase enzyme. And here you can see the in vitro after the construction of a recombinant DNA harboring the foreign DNA along with the background of the bacteriophage DNA, you can subject it to in vitro packaging, where the recombinant viral DNA particle will be mixed with the proteins of the lysate that will help to package the or assemble the recombinant DNA into the capsid or the viral DNA head system. So these are all examples of phage vectors with a varying capacity of insertion. So we have on an average 20 to 25 kilobase per insert capacity that can be put for as an insert to clone in the bacteriophage based vector system. The next vector system is Cosmids. So these Cosmids, as the name implies, they are a derivative of plasmids. So Cosmids are basically plasmids which possess the Cos site from the phage DNA. So plasmid which possess Cos site are called as Cosmids, and they are reported by Collins and Hahn in 1978, and they are used to clone larger DNA fragments by exploiting the same phage in vitro packaging system. So we make use of the plasmid. In addition, we are making use of the Cas site that's been transferred from the viral system for efficient in vitro packaging. So Cosmids are available in large numbers from different systems, and they have a carrying capacity of approximately 30 to 50 kilobase per means they have a capacity double the size of the phage vector systems like lambda phages, and they have a capacity 5 times better than the plasmid DNA systems, plasmid DNA vector system, because plasmid DNA can averagely they can have an insert capacity of around 10 kilobase per, and here in case of Cosmids they have an insert capacity of about 50 kilobase per in size. So these Cosmid they essentially combine the elements of a plasmid and the lambda systems. Here the Cosmids can be extracted from a bacteria and they can be mixed with the restriction enzyme. Once the Cosmids are cleaved, they can be mixed with the foreign DNA to produce a recombinant DNA, and the recombinant Cosmids which possess the foreign DNA can be packaged like that of a viral DNA using the in vitro packaging systems. So recombinant plasmids are injected into the bacterial cell where the Cosmid will arrange into a circle and they replicate very much like a plasmid. Although in case of lambda DNA, we have seen the lambda DNA being present as a linear DNA. Here they can replicate like a plasmid because they encircle themselves and form a circular molecules. So they can be maintained and recovered just like plasmids. The fourth vector system is Phasmids or Phagemids. These Phasmids or Phagemids are DNA-based cloning vector systems. They also possess both bacteriophage as well as plasmid properties. And these plasmids, they carry an ORI region or origin of replication region from the bacteriophage F1 in addition to the bacterial origin of plasmid ORI region. So in the sense, they have a two choice of host system to multiply. One is in the viral vector system, virus system. The other one is in the bacterial system because they possess the ORI region from two different host cells. One is from the bacteriophage, the other one is from the bacterial system. So due to their having a genetic sequence that signals for packaging, they can be easily used for in vitro packaging like that of any phage vector systems. And normally we use these phagemids or phasmids in molecular biology technique called as phage display to study the protein-protein interactions and receptor-ligand combinations. Next, we will see a type of vector called as shuttle vectors, as the name implies, shuttling between the host systems. So these vector, usually a plasmid is capable of propagation in two different host species because they will have a combination of signals for amplification in two different host systems. They can be manipulated in E. coli. That's the biggest advantage we have. They can be easily manipulated in the E. coli, but then they can be used to be amplified or used to produce a foreign particle or recombinant protein in a similar eukaryotic system like yeast. So they can be shuttled between prokaryote and eukaryote. That's why we call them a shuttle vector. Say, for example, here we have given an example of shuttling between the Baker's yeast, Saccharomyces cerevisiae, and E. coli, or in different species of bacteria, you can make a shuttle between E. coli to Rhodococcus erythropolis. So these vectors are also capable of multiplication between bacteria as well as mammal systems. Some of the adenovirus shuttle vectors are good example of propagation in both bacteria as well as mammalian systems. Almost all commonly used vectors that are derived from the plasmids, Saccharomyces cerevisiae, are basically shuttle vectors. And these yeast shuttle vectors will contain two different components. One is a bacterial component, E. coli component, which possesses the origin of replication from E. coli and a selectable marker gene, preferably an antibiotic marker genes, which will produce an enzyme capable of degrading the beta lactam containing antibiotics. The second component in the shuttle vector system is yeast component. The yeast component encompasses the ARS region, that is autonomously replicating sequence, a yeast centromere, and a yeast selectable marker, that's uracil3, gene that encodes the enzyme for the uracil synthesis. The next type of vector system is expression vector system. As the name implies, expression construct. It is also referred as expression construct or expression vector to produce the foreign protein in host systems. So these vectors are primarily used as an expression system to get the cloned DNA or a gene of interest to produce the recombinant proteins. So what we do is here we introduce the genes into a vector system. A specific gene is introduced into the target host cells, and they commandeer the cell's mechanism for protein synthesis to produce the foreign protein that is encoded by the gene of interest. These vectors are commonly used as a basic tools in biotechnology to produce recombinant proteins. The most commonly used bacterial expression vector is PET vector that was originally supplied by the Novagen company earlier. And these PET vectors have the biggest advantage in the sense that 50% of the total protein that's produced by this vector system is recombinant protein compared to the total cell protein of the bacteria. Some of the yeast expression systems include the plasmid pic series vectors, that is using Pichia pastoris as an expression host systems. And based on the TI plasmid, we have plant expression vectors that includes the P. cambia and as well as PUH. So there are series of P. cambia vectors available that is used for transforming monocots as well as dicot plants. P. cambia 1300, 1301 series or 2300, 2301 series are routinely used in the plant genetic engineering to create transgenic plants by transforming the genes of interest coding for a particular trait. So the expression vector must essentially contain genes that are necessary for gene expression. In the sense to transcribe a cloned gene, it needs a promoter region. So the most commonly used promoter is the T7 lag promoter in a bacterial expression system, such as it also possesses the ribosome binding site, RBS site, and a start codon, a termination codon, and a termination sequences for terminating the transcription event. So there are differences in the machinery for protein synthesis between prokaryotes and eukaryotes. Therefore, the expression of expression vector will have elements that are appropriate for the chosen host. Say, for example, the prokaryote expression vector should definitely have a SD or a signed Algorno sequence at its translation site, which will be helpful for binding of the ribosomes to initiate translation process. Whereas in case of eukaryotic expression vector, they would have the complement or an analog of the SD sequence of prokaryote that's called as Cossack consensus sequence in the eukaryote that helps in binding of the ribosome to initiate translation process. The next category of vectors are artificial chromosomes, which include a bacterial artificial chromosome, which is a DNA construct which is based on the functional fertility plasmid, F plasmid, that is used for transforming and cloning bacteria, usually in E. coli. So these F plasmids play a crucial role because they contain genes for partitioning that promote even distribution of the plasmids after the binary fission or a bacterial cell division. So the bacterial artificial chromosome BAC vector have the insert carrying capacity of about 150 to 350 kilobase pair. That's a larger size inserts that can be potentially used for cloning larger genomic fragments, constructing genomic DNA libraries. So the BAC vector or a bacterial artificial chromosome vector contains some of the gene components, which includes the ORI region, that's origin of replication for replicating the DNA, as well as they have rep E to F factor, EF factor for regulating the copy numbers during the multiplication. As we said, it contains the partitioning plasmid DNA. PAR A and PAR B are the genes that are present in the F plasmids that will help partition the F plasmids to the daughter cells during the division. So they will ensure the stable maintenance of the bacterial artificial chromosome while partitioning the complements to the equally to the daughter cells. For easy selection, the selectable marker for antibiotic resistance is also present, and some bacterial artificial chromosome possess the LAC-Z gene, which codes for LAC-Z gene codes for beta galactosidase that aids in blue-white screening of the recombinants. The T7 and SPs are phage promoter regions, which are used for transcription of the cloned genes. Basically, we place the foreign gene under the transcriptional control of T7 and SP6 vector systems. So here you can see this is a back vector which possesses the cloning site here, the F plasmid, and an origin of replications for independent replication. When this back vector is subjected to digestion within restriction endonuclease, it cleaves the cloning site. So the cloning site is cleaved and they make the large cut open of the vector where you can clone a large foreign DNA into this uh, plasmid bacterial artificial chromosome regions. As you said, it contains about 350, approximately 350 kilobase per long DNA fragments can be cloned in the back vector. So you can ligate them to produce a recombinant back vector, which can be subjected to electroporation to get mobilized into the host system, and they can be subjected to selection by using an appropriate antibiotic system for screening for the presence of recombinant proteins. So the colonies that appear white are supposed to be carrying the recombinant bacterial artificial chromosomes. The next artificial chromosome is YARC, that's yeast artificial chromosome, which has the insert carrying capacity of about 100 kilobase pair to up to 3000 kilobase pair. This is 10 times larger than the back system itself, or 300 times larger than the conventional plasmid system we use for manipulations. So this was first, the YARC system was first described by Murray and Sastek. It's an artificially constructed chromosome which contains a centromere, telomere, and an ARS element from the yeast sequences. They are required for replication as well as preservation of the YARC vector system in the yeast cells. And these YARC vectors with a larger insert capacity are very much useful for physical mapping of the complex genomes as well as cloning of larger genes in the library DNA library constructions. So these yeast expression vectors based on the yeast vector systems include the YARC vectors, yeast integrating plasmids, yeast episomal plasmids. They have advantage over and above the bacterial artificial chromosomes which we used to produce the eukaryotic recombinant proteins, because the recombinant proteins that is expressed from the eukaryotic gene, normally in a prokaryotic express system we have a problem with post-translational modification because of poor recognition by the host bacterial host system. Whereas if you express in the yeast-based vectors, the post-translational modification will be best compared to the bacterial host system. So a plasmid-derived ORI and an antibiotic-resistant gene allow the YARC vector to be easily amplified, and they can be subjected to screening in a bacterial host system. PRP1 and URA3 genes are included in the YARC vector because they efficiently provide the selection system for identifying the transformed yeast cells. The TRP1 codes for phosphoribosyl anthranilate isomerase, and URA3 codes for orotidine 5-phosphate decarboxylate enzyme that is used for a selection system in identifying the transformed yeast cells. So this picture depicts the yeast artificial chromosome P-YARC2 vector possessing the centromere, the yeast origin region, the yeast selectable marker URA3, a telomeric region, and here you can see the origin of replication, which is from the bacterial origin for shuttling between the bacterial as well as yeast host vector systems. The third artificial chromosome is PAC, which is a artificial chromosome derived from the P1 phages. So a PAC is a DNA construct. It is a derivative of the P1 bacteriophages, and they combine the capability of the bacterial artificial chromosomes. So they can be efficiently used to clone the DNA fragments of 100 to 300 kilobase per size. On an average, you can clone about 150 kilobase per of DNA, foreign DNA in the PAC chromosomes, artificial chromosomes, and they can be multiplied in the E. coli cells. So this P1 phage was developed as a cloning vector by Nat Sternberg and colleagues in 1990s. So the application of these P1 artificial chromosomes is very much helpful in construction of genomic DNA libraries of human, mouse, and even other organisms. And this was very much used in the human genome projects. And these libraries, DNA libraries, serve as template for larger gene sequencing and genomic analysis for analyzing the specific functions exhibited by the different genes of complex organisms like plants and animals. And these artificial chromosomes are capable of expression and facilitate gene expression of the cloned DNA producing recombinant proteins. As a summary, we will be discussing on the maximum insert DNA capacity of different cloning vector systems, which you can see the lambda phage has about 25 kilobase per insert cloning capacity, whereas the Cosmids, which is derived from the Cos site of a phage and integrating the components of a plasmid, bacterial plasmid, has 35 to 45 kilobase per insert capacity. The P1 phage, an unedited P1 phage, is basically having an 100 kilobase per capacity, but if the P1 phage is combined with the bacterial artificial chromosome or modified to have a pack system, P1 phage based artificial chromosome, then you can have an increased capacity of 100 to 300 kilobase per inserts. The bacterial artificial chromosome has a capacity of about 300 kilobase per, whereas an yeast artificial chromosome has a capacity of about 2000 kilobase per, up to 2000 kilobase per DNA can be cloned. So that's about the types of vector system we deploy for recombinant DNA technology to transfer the gene of interest for producing recombinant DNA molecules. Thank you.